And welcome to the, what month is it? September Neuroprosthesis Seminar. It is my honor to introduce Rob Turner. He got his bachelor's and PhD at the University in Washington, studying cellular and molecular biology as well as physiology and psychology. And from there he went to Emory and worked with Malin DeLong, working in movement disorders. And then he transferred to UCSF, did his early faculty work there. And in 2007, he went to the University of Pittsburgh and is now a full professor in neurobiology and biomedical engineering. He is one of the premier researchers that has been painstakingly slogging through trying to understand the details of these circuits that are controlling our movements. Very few people have the patience to do what he does. And he's been trying to understand basal ganglia, cortical, the whole motor control loop, both in the normal state and in diseases, and then, understanding why what we're doing to it to try to cure people actually works and sometimes doesn't work. He's got a long history of very successful NIH funding with R01s, R21s, P grants, U grants. He's been on numerous review panels and reviewers for various journals. And today we get to hear, he's going to tell us the facts about how DBS works. We've all been eagerly waiting to hear this. So welcome, Rob.
Thank you so much, Dawn. This is a real honor. Thank you so much for inviting me, and it's just a short drive down the turnpike. So it's wonderful to tell you some of the work that we're doing about some of the work. And I hope that I don't want to overpromise with this title. I'm sorry. Maybe those of you who expect that it'll be all figured out by the end of this hour, you can leave now. Sorry to disappoint. But I think that we have made progress in understanding deep brain stimulation. And as Dawn mentioned, it seems like incremental progress. Is the glass like an eighth of a bit full or mostly empty? Well, we'll see by the end of this what I have to say. These are the main points that I would like to convey to you today, and I hope that some of the data that I show you convinces you that there's some substance to these 3 points. The laser pointer's battery's out here, so you can see my pointer here, right? So we have pretty good evidence that deep brain stimulation blocks the transmission of what I'm going to tell you is what pathologic activity is. So I'm going to spend quite a bit of time giving you illustrations of what we mean by pathologic activity. And we think that deep brain stimulation is working by blocking the transmission of this abnormal signal. And how does DBS do that? How does it block? Block sounds so simple, but how does this work biophysically? We have some evidence, modeling evidence primarily, that this blocking effect is by the induction. It works by induction of forms of short-term depression in neurons, neuronal elements that are being stimulated. And there is a hypothesis in the field that DBS works by producing what is called an informational lesion, so that it doesn't actually lesion tissue, but it blocks the transmission of information through the stimulated tissue. And we have evidence that suggests that that's not completely true, that informational lesion hypothesis, it needs to be taken with a grain of salt or reinterpreted. So that just to make sure we're all on the same page as far as deep brain stimulation, I'm sure this is old news to most of you. DBS is a form of deep brain stimulation that's permitted by a neurosurgeon lowering an electrode deep into the brain to stimulate areas mostly in the basal ganglia for movement disorders. Other areas of the brain are also being used. It's a neurosurgical procedure, and the leads are implanted deep in the brain. This is a transmission X-ray showing the leads that are deep in the brain, and the leads are then led subcutaneously to a pulse generator that looks something like this. It's implanted below the clavicle, and the working end of the probe looks something like this. This is a standard probe. New generations of probes are out now, but this is the standard probe from Medtronic, and these are the stimulation contacts. And the characteristics of standard deep brain stimulation, what's being used widely in the field now is the stimulation is a short duration electrical pulse. It's less than a tenth of a millisecond, so very short electrical pulses. The stimulation is at high frequency, over 100 hertz stimulation. This is actually not so true anymore. I'm sorry, it says constant voltage. So for many years, the pulse generators were voltage controlled stimulation. The new generation is now current controlled stimulation. And the stimulation is delivered continuously, 24 hours a day, 7 days a week, continuously, high frequency stimulation. And some of these characteristics, for those of you who study stimulation of neuronal tissue and knows about the biophysics of that process, can recognize that some of these characteristics suggest that deep brain stimulation is having its principal effect on fibers, large myelinated fibers, and not on cell bodies. For instance, the short duration of the electrical pulses makes the stimulation more effective at driving fibers of passage or terminals. Okay, and so DBS is used, its use for some disorders, movement disorders, is very well established, and it's starting to be used for explored. There is work exploring its utility for an ever lengthening list of other disorders, including substance abuse, tinnitus, cognitive dysfunction associated with Alzheimer's, even PTSD, and as you know from work being done here, it's being explored as a potential therapy for stroke. And it wouldn't be surprising for some of us to start asking the question, well, so is deep brain stimulation sort of like a hammer? If the only tool you have is a hammer, then everything starts to look like a nail. And this idea was stated some time ago by Maslow with reference to the use of Thorazine in psychiatric clinics, but maybe we should ask the question whether deep brain stimulation is so effective for some disorders, or is it fair for us to start thinking of it as, well, it works so well, let's try it on some other disorders. But by the end, I hope that I convince you that maybe it's reasonable to consider it as a potential therapy for many disorders, because many brain disorders can have pathologic activity that DBS is effective at reducing. Okay, so let's start out with a historical perspective. Why even try DBS? And this will get to the question of what is pathologic activity, and why did people start using deep brain stimulation, and that will give you some insight to the kind of historical biases and our understanding or our hypotheses about what is the the mechanism of action of deep brain stimulation. And I'll go on to tell you later about local effects and some of our experiments looking at the local effects of deep brain stimulation and then system-wide effects. Okay, and so this is just a cartoon of a parasagittal section through a macaque brain. A lot of the parts of the macaque brain are similar to that in humans. I'm sure you recognize that. And the loop circuit that travels through the basal ganglia is the circuit that dysfunction of this circuit leads to the symptoms of Parkinson's disease. And dopamine plays a highly significant role in Parkinson's disease. And we like to summarize this circuit, loop circuit, with a wiring diagram, something like this. And there's a lot of internal complexity in this wiring, but the primary point is that there's a bottleneck in the output from the basal ganglia. And this nucleus is abbreviated GPI as the internal globus pallidus. And this is the primary output nucleus for the basal ganglia, especially the part of the basal ganglia that controls movement. And the GPI projects to thalamus with inhibitory fibers. So these are GABAergic fibers that project to thalamus that projects back to cortex. And so the basal ganglia receives input from motor cortex and projects back to motor cortex by way of the internal globus pallidus. We know that the symptoms of Parkinson's disease, the cardinal symptoms, are caused by loss of dopamine deep in the basal ganglia. And the loss of dopamine leads to symptoms, the cardinal symptoms being akinesia, bradykinesia, that's slowness of movement, limb rigidity, and postural instability, a number of other symptoms, but these are the principal symptoms, motor symptoms of the disease. And so when we're trying to understand, well, so what is the connection between loss of dopamine deep in the brain, deep in the basal ganglia, and these global symptoms? There's some causal cascade that leads from the pathology to the symptoms. And that's, instead of just thinking of it as a causal cascade, let's call it pathophysiology. Okay, and a long train of experiments have shown that the part of the pathophysiology, a key part of the pathophysiology, is abnormal activity in the neuronal activity in the internal globus pallidus, in the GPI. And that is projected out, transmitted out to the thalamus, and appears to disrupt function of the thalamus and cortex. Okay, and so what are some of these abnormalities in activity what I'm talking about? So I call it pathologic activity, and some of the abnormalities in activity, there's a long list of abnormalities of the neuronal activity recorded from this nucleus, but they include increased firing rates of neurons. The firing of these neurons is bursty, which means that the action potentials are clumped together in time. The activity is not just bursty, but also oscillatory, so it's rhythmic. And that rhythmic, bursty activity is synchronized between neurons. So neighboring neurons fire together. There's some other, as I mentioned, there's a relatively long list of abnormal features to this activity that make it quite different from what is seen in a normal animal. And so for Parkinson's disease, we can say that pathologic activity, this term that a lot of us in the field are using now, is a big part of the pathophysiology. So instead of just saying some vague term, pathophysiology, we can put pathologic activity as being a critical step in the causation of symptoms. All right, so what do I mean by pathologic activity? I'd like to give you just some examples so that this is a dynamic process. We record from neurons using extracellular electrodes that are lowered deep into the brain. We can do this in human patients during DBS surgery. We also do it in animal models of Parkinson's disease. And so what I'm going to show you here is the action potentials generated by one neuron in the globus pallidus. And this is a recording that is taken from the OR in a collaboration that I did with Phil Starr when I was at UC San Francisco. And one of my graduate students wrote this up into a paper. And so what we're doing here is we're showing the action potentials triggered on a threshold here. And so you'll just hear and see the action potentials as they're picked up by the electrode. Let's see. So the rhythmicity of that activity should be pretty obvious to all of you. One of the interesting things about being a neurophysiologist is you realize how good the ear is at doing analysis that is not so obvious to the eye. But even here, this is the same pattern just shown in 2 dimensions, time, voltage, and you can see the rhythmicity of that activity. And we can do a spectral analysis of that just picks out the frequencies. And here we go. In the spectral analysis, it measures the power at different frequency range measures. And so we see a very high peak around 5 hertz, and another peak around 10 hertz, and these peaks reflect the power in that spike train. And this type of pattern is never seen in neurologically normal animals. Okay, so this is one example, and these frequencies are close to tremor. The frequency of tremor, the 4 to 8 hertz tremor that's seen in Parkinson's. And here's another example. This may be a little bit more subtle, and I'd like you to listen more than watch, because I hope you can pick up a frequency, dominant frequency in the activity of this neuron. And this neuronal activity was recorded from a monkey model of Parkinson's disease. We gave the monkey MPTP, a neurotoxin that's specific to dopamine neurons. And again, this is recorded from the globus pallidus, this output nucleus of the basal ganglia. Do you hear that rhythmicity? It's much higher, but there is a rhythmicity there too. Okay, and if we plot it again, voltage versus time, I hope you can see the rhythmic bursting of that activity, and the spectral analysis confirms that there's a high peak at a frequency that's higher than tremor frequency. It's about 15 hertz for this neuron. And this is in the beta frequency range. And beta frequency oscillations have been strongly implicated in the pathophysiology of Parkinson's. And you'll be hearing a lot more about that as the talk progresses. This sort of beta frequency oscillatory activity isn't seen just in Parkinsonian monkeys. We also see it in humans. This is data that we collected from the OR. Phil Starr was a neurosurgeon in this study. And if we do a spectral analysis of this spike train, here we see a very nice spectral peak right at 20 hertz, which is right at the middle of the beta frequency range. And just as a comparison, so how do we know that this is abnormal, that this is pathologic? Well, this is what the spectrum of all GPI neurons looks like in neurologically normal animals. It's flat. There's very, very little oscillatory activity in a neurologically normal animal. Okay, so this oscillatory activity is a very important aspect of the pathologic activity. I also mentioned synchrony. Well, so synchrony, the synchronized firing between neurons, is something that I can't show a movie of. At least I haven't figured out how to do that yet. But we can take the spiking activity of a pair of neurons recorded from using separate electrodes, different microelectrodes in the globus pallidus, and then we look at the coherence in the firing between those neurons. And so coherence is how well correlated is the time of spiking of those neighboring neurons. And so coherence is something like a regression analysis. So it can go to the peak coherence would be 1, right? And so this peak in the coherence that we're seeing, and it's frequency-specific measure of the how well correlated this activity is. And so we see a peak for a single pair of neurons. We see a peak that's around 15 hertz for this monkey. And we look at the coherence for pairs of GPI neurons. There's high coherence across a range of frequencies, but that coherence includes the beta frequency range. We also see the same kind of coherence, similar kind of coherence when we record in primary motor cortex. And so here's an example pair with a strong peak, a pair of neurons recorded from primary motor cortex. And in that population also, there's a very high prevalence of coherent firing in the beta frequency range. Okay, so why do we think that this pathologic activity is important in the symptoms? What makes us think that, with some confidence that that pathologic activity I've been illustrating really does lead to the symptoms? Well, the simple answer is we know that pallidotomy works. Before the field discovered deep brain stimulation, Parkinson's patients, they took Cinemet, dopamine replacement therapy, but after years of the dopamine replacement therapy, many patients started suffering side effects, unwanted and reduced benefit from Cinemet. And neurosurgeons look back in the history of their work, and they just were reminded that lesions of the GPI lead to a reduction in symptoms. And so there was a resurgence in the use of pallidotomy in the years immediately before the use of the discovery of deep brain stimulation. And so a lesion of these neurons that have this abnormal activity leads to a significant reduction in the severity of symptoms. And there's no controversy about that. I, at least to my mind, I have heard nobody disagree with that. There's a huge literature that supports the efficacy of a palatal lesion at reducing symptoms. And so that, in my mind, that's all the evidence that I need to convince me that this activity is leading to the causation of the symptoms. Okay, well, so now let's jump up to the current day, and we've got deep brain stimulation. And so we're stimulating GPI electrically. And we know that does reduce symptoms, or, I haven't shown you evidence, but just take my word for it. We know that it works, right? But what's it doing to the pathologic activity? What's its effect here? And you might be, there's reason to assume, well, the end effect of deep brain stimulation, the effect on symptoms, is just like a lesion. So it would be tempting to just say, well, it's producing a lesion. It's somehow functionally lesioning this nucleus. But it's electrical stimulation. And how many decades of work do we have that make it very clear that electrical stimulation of neuronal tissue drives neurons? It doesn't inactivate neurons. So that raises questions of what are the local effects of DBS? And then how are those local effects translated into global relief of symptoms? Okay, and so let's talk a bit about the local effects. And does DBS work like a lesion? And by lesion here, I really mean inactivation, like an ablation. And the key scientific results is in this paper from Jerry Vitek's group. And most of this data was collected when he was at Emory University, and I overlapped with Jerry there. And Hashimoto and all recorded from neurons in the GPI, internal globus pallidus, while they stimulated the subthalamic nucleus, which is one synapse away. And the subthalamic nucleus provides very strong glutamatergic excitation of the GPI. And what they saw in their recordings is that, no surprise, you electrically stimulate these glutamatergic neurons that excite GPI neurons. If you stimulate in the subthalamic nucleus, it will drive activity in the GPI. And so this is during 2 Hertz stimulation. This is the shock artifact. The squiggly lines are action potentials, so there are multiple traces overlapping each other, but you can see this cluster of action potentials that follows that shock, and then another cluster of action potentials at a longer latency. If they increase the frequency of stimulation, you can see 2 shocks in this figure because the time between shocks has gotten shorter. But still, even at this high frequency, these neurons fire. Their firing is entrained to the stimulation. And even up to 157, 160 hertz, you can see an entrainment of the firing of that neuron. The neuron's activity is not blocked. There's not a depolarization block process going on in this neuron. And that was true for the population of neurons. And so across many neurons that they recorded from 2 animals, the firing rate of these neurons was actually elevated during DBS. So much for the lesion idea. Okay, and so we did some recording. Kevin McCarran, when he was working in my lab at UCSF, did some recording work. He recorded in the globus pallidus during stimulation, DBS, in the globus pallidus. So he's recording from neurons that are about a millimeter away from the stimulating electrode. So this is a slightly different picture than what Hashimoto and Vitek did. But we've got, our results were fairly similar to what Hashimoto and Vitek found, in that stimulation, here's the time of a shock onset, and this is the activity of a GPI neuron, and you can see that this neuron is driven at very short latency with very high fidelity and little jitter following the shock. And this is many traces overlapping each other. And the interesting thing that we saw is that this looks very much like an anadromic activation. So it's a direct activation of this neuron there were a few times when the shock failed to drive this neuron to make an action potential. And at the times when that driving effect failed, the shock was preceded by a spontaneous spike. And this is very much like what we see normally in neurophysiology. We see collision between a spontaneous spike and the stimulation-evoked spike. More evidence, not all neurons fired exactly like this, but we found results that were very similar to what Hashimoto and VTech reported. And so our results agree with their description that DBS is exciting the neuronal tissue, not inhibiting it or lesioning it. Okay, so if we want to go into more depth and saying, well, okay, so deep brain stimulation, we're stimulating neuronal tissue, and so what are the key parts of the brain, the key neuronal elements for producing therapeutic benefit? Well, so the first thought when we're stimulating in gray matter is, well, there are neurons in the gray matter nearby the electrode contact, and we could be driving the spiking activity in those cell bodies. But as I mentioned, the most effective form of stimulation is short duration pulses, and other features of DBS suggest that it's affecting fibers, and that the driving of fibers is the important part of the therapeutic part of deep brain stimulation. And it turns out that there are a good number of fiber pathways nearby the subthalamic nucleus. And those fiber pathways include, but not exclusively, they include a pathway from globus pallidus, GPI, to thalamus. It doesn't actually follow this path, but it's nearby the site of stimulation. There's a projection from cortex. Terminals, neurons project into the subthalamic nucleus from cortex. And so these terminals are nearby the site of stimulation, and so we could get antidromic activation that travels up these axons, corticothalamic axons. And then there are other pathways also. So the GPE projects into the subthalamic nucleus, and so we could get antidromic activation of those fibers also. And then corticospinal fibers are nearby the subthalamic, and perhaps some of the therapeutic benefit could come from driving of these fibers also. So how do we tease this apart? How do we determine which fiber pathway is most important, and how does excitation of a fiber pathway, how does that translate to benefit? Well, we took a look at some of the effects of deep brain stimulation, and that they may provide a clue to one possible mechanism of how DBS is changing the tissue, the way the neuronal tissue works, and it could, in a way that could lead to therapeutic benefit. Okay, and so in our recordings, this is data that we collected, that Kevin McCarran collected. So it's stimulation very near, recording very near a stimulating electrode in the GPI, in the globus pallidus. And this is an example neuron, and what I'm showing the firing rate of this neuron relative to the cell's baseline firing rate. And so it's a slightly different way of showing it, but the main point is that during the first 10 seconds of stimulation, there's a strong driving of the neuron, followed by a reduced firing, and then maybe a little blip of increase and another decrease. So we see this modulation of the firing pattern of the neuron. And notice the time scale here. So we're stimulated at high frequency, and the next shock is right off the screen, off this figure. Okay, so this is the pattern that we see the effect of DBS on this neuron's firing in the first 10 seconds. In later segments of time, the effect changes. It changes over time, so that this early excitation is reduced, and that this later what appears is a reduction, a cessation of inhibition, it becomes an increase in firing. It becomes even more prominent later during the 20 to 30 seconds after stimulation onset. Well, that's an interesting evolution of the effects of DBS across a fairly short time course. This is not hours of stimulation. This is seconds, tens of seconds. Well, this phenomenon has been reported by others as well, and this is from a study, McCarran and Bargad in Israel. They recorded in the GPI and stimulated the subthalamic nucleus, and their result is shown here in raster form and then also in histogram form. And so in this raster, the top row of tick marks shows the firing of a neuron around the time of the first stimulation. And successive stimulation, the effects of successive stimulations are shown in later rows. And so this speckling here is that shows each speckle shows the time of one action potential. And so across successive shocks, you can see a slow increase in the latency of the response of this neuron. It's responding with an increased firing, but it becomes later and later and slightly smaller. And that is borne out looking at these histograms. So the first histogram is taken from the very first few stimulations, and the red histogram here is taken from the last few section of the stimulation. And it again shows that there's an evolution of the response. Okay, and so Robert Rosenbaum worked with, did a postdoc with Jonathan Rubin and I, and he saw these results and thought, you know, this is an interesting result that might actually be giving us some clues to a biophysical mechanism for why DBS works, why it's producing benefit. Just to hammer home the fact that this sort of slow evolution of the effect of DBS is common. We more recently did another study where we recorded in the globus pallidus and did stimulation in the subthalamic nucleus, and across a large population of neurons, we saw during the first 10 seconds of stimulation a large increase in firing. This dotted line is the baseline firing rate of neurons, normalized, and this is the response during the first part of stimulation, first 10 seconds, and after 50 to 60 seconds of stimulation, the response is dampened. It's quite a bit smaller. So Robert thought, he did some reading in the literature, and you know, there are some features of neuronal firing that we really should take into account when we're trying to understand the effects of DBS. And some of those effects include forms of short-term depression. There's a short-term depression in the reliability of an axon to conduct action potentials. There also is a short-term depression process in synaptic conduction, just the process of vesicles fusing with the presynaptic terminal and releasing neurotransmitter. There's a short-term depression process that happens in synaptic release. And so Robert modeled those processes in a detailed way based on the best evidence that we have for measures of how axonal failure happens and synaptic failure happens. And his conceptual model goes something like this. So if you can think of this as being a subthalamic nucleus neuron, and we're recording right at the axon hillock from that subthalamic nucleus neuron, and also doing deep brain stimulation nearby. And so without DBS, this neuron is producing action potentials. just naturally occurring action potentials. And then DBS, when DBS turns on, the DBS is also producing nascent spikes at the axon hillock that are induced by the DBS. So we get laid on top of each other what the action potentials that would be happening naturally, and also action potentials that are being elicited by DBS. Okay, and so then what happens in the downstream structures? Well, the axon hillock produces what Robert calls nascent axonal spikes, but not all of those spikes are conducted along the axon. There's failure in conduction along axons, and that failure rate depends on the firing rate of action potentials along that axon. So there are failures in the conduction of some of these nascent action potentials to be conducted along this axon. And that failure rate is going to attenuate some of the effects of DBS. Some of the spikes that are nascent spikes at the axon hillock induced by DBS, they will not be transmitted along the axon. And then there also is a failure at the synapse, a failure to conduct between pre- and postsynaptic membranes. And so that failure will also lead to reduced transmission of some action potentials. And you can see some examples here in this model. So if we wire up a model of a population of these subthalamic nucleus neurons spiking like this, and GPI-like model neurons that are receiving input, and these two short-term depression processes are included in the model, then we can see that this pathway from subthalamic nucleus to the GPI is unable to transmit beta frequency oscillations. And so this figure shows the ability of that pathway to transmit beta frequency oscillations. And so this is showing frequency, and think of the Y scale as being the ability to transmit a signal at different frequencies. And so we have a strong beta oscillation being transmitted along this pathway, and with the induction of these two forms of depression, that transmission is attenuated significantly. Okay, and so an important part about this is that something that's it's important to keep in mind with this model is that what we're not saying that there is an attenuation of the firing rate in the GPI neurons, or even an attenuation of the rate of action potential conduction along this axon. It's just that there are more failures because we're trying to introduce more spikes. And so this plot shows a model neuron that is receiving beta frequency oscillations coming in. And when we turn on deep brain stimulation, we see a short-term strong response, similar to what we see in the real data, strong response followed by this short-term depression and failure of this neuron to follow that same beta frequency oscillation. But the neuron, this postsynaptic neuron, is still firing at a high rate. Okay, so this is a possible mechanism, and we need to do a lot more experiments to test this hypothesis. And the results that I'm showing you are results from models. This is empirical data, but the model, Robert's model, fits the data that is available fairly well. We need to test the model more to see if it really, yes, sure.
The red line is the electrode at the top. And you're driving the cell with that electrode there, or are you driving the synaptic input to that cell?
That's a good question. that's a good question. And so the way the model is designed is that the nascent spikes induced by DBS are conductances. They're EPSPs, or depolarizations at the axon hillock in this presynaptic cell. And so Robert didn't model, provide any more detail as far as how the inputs arrive. How are these inputs produced? He just modeled them as EPSPs.
So if you had a stronger synaptic input, that failure would probably not occur that you're looking at in here.
Yes.
Okay.
Yes, that's right. Yeah. Well, no, there's still failures, right? So it depends on which you mean, right? So there is a certain failure rate, and it's stochastic. There's a failure rate in the conduction of action potentials along an axon. This happens all the time. It's just that the failure rate increases when we're trying to push more action potentials along that axon. And the same with synaptic conduction. The failure rate increases the more action potentials we try to push across the synapse.
But if there is long-term depression, how you can explain that the fire rate does not go down? Wouldn't be this better explained by desynchronization? So you have synchronized activity, you stimulate like this, and that produces just greater activity that desynchronize those. It's like a switch in the profile of the activity rather than long-term depression.
And so, but what is the source of desynchronization? What causes the desynchronization?
Well, yeah, that you put a lot of an amount of stimulation at high frequency on this input.
Yeah. So I don't have the other figures. So Robert made the same model. He has a result that shows the same model, but without depression. So the model has firing of the STN and introduces more action potentials. This firing is beta oscillating firing and introduces more action potentials. But when it's not depressing, when he takes out the depression parts, you get the beta oscillations before DBS, you turn on DBS, and you still get beta oscillations, just at a higher frequency. And so at least in his model, the increased firing rate doesn't block the transmission of beta. It is not, so it's just there are more action potentials, there's a higher baseline, but the transmission of oscillatory firing is still there. Okay, as I said, we want to test this. And if you have ideas of how to test this, I'd love to hear. A colleague of mine at Pitt is doing some slice work that we hope that will help. And we have some experiments planned for monkeys that we hope will help test that. Okay, so now a little time remaining. Let's talk a bit about system-wide effects. And so what features are normalized? And just to skip to the good part, we have a good bit of evidence that reduced baseline firing rates, that is increased firing rates is a pathologic feature, or feature of pathologic activity. But that is not the key change in activity that is leading to reduced symptoms. Change in burstiness is probably also not the key change. But we have oscillatory firing and synchronized firing between neurons. What about that? And so this is the last little movie that I have. And so this neuron you heard a bit of from before, but we also studied it during DBS right nearby the site of stimulation. We recorded from this neuron nearby the site of stimulation. And so, I should. Sorry, I should have told you. So you'll see a red bar up here, and the red bar comes on when the stimulation starts, DBS starts. Let's see. Okay, so there is a change. DBS is inducing a change. There's a short term, right after we turn it on, the neuron is inhibited, but then afterwards there is a reduced rhythmicity. And you might be able to see that in the comparison of off versus on DBS. It's very obvious in the spectrum. There is still a peak at that 15 hertz frequency, but it's much reduced compared to the off DBS condition. Okay, and we saw this across. Not only did we see a reduced oscillatory activity in individual neurons, but we also saw a reduction in coherence across pairs of neurons. And this is pairs of neurons, so this should be familiar to you. You saw this peak in the example pair and increased coherence at multiple frequencies, and the red bars show the change in coherence with DBS, and there's this large drop in the frequency of coherence in GPI, and also we saw this similar kind of result in the motor cortex. Recording from pairs in motor cortex, there's a large drop in the beta frequency coherence.
But, I noticed in the 2 times that you have presented similar thing that the theta activity is not affected, yeah? The tree is,
yeah.
Why is that? Why is it not affected?
I don't know. Yeah, and there's quite a bit of theta activity, but it is, yeah.
And this is not normal.
Oh, no. No. So the incidence of coherent activity is virtually zero in the normal monkey, neurologically normal monkey.
And despite you don't affect these, there is improvement.
Yes. These animals show therapeutic effect of DBS. Reduced rigidity, more movement. Mm-hmm. Yeah. Okay, so I'm going to just skip ahead. We're out of time. So our evidence supports the idea that oscillatory firing, DBS works. It does reduce some aspects of pathologic activity. And some of the things that it reduces include oscillatory firing and synchronized firing between neurons. Okay. So let's see. So I'd like to show you just a little bit of data that we have on this idea of the informational lesion. So can we call this effect, even the short-term depression effects that I showed you, is it blocking the conduction of information through the basal ganglia? Well, it depends on what you mean by information. And so Andrew Zimnick worked in the lab before going to graduate school, and he recorded in the globus pallidus while stimulating DBS. And he did this work in neurologically normal animals, performing a simple touch task. So they reach from a home key to a target, one of 2 possible targets, the monkey gets a reward, a little bit of food for doing the task correctly. And no big surprise, neurons in the globus pallidus have firing that modulates with movement. And so this is the average activity of a single GPI neuron aligned on reach onset. And so there's a big reduction in firing of this neuron around the time of movement onset. All right, so then we turned on deep brain stimulation in the subthalamic nucleus. And this neuron had significantly elevated firing when the animal's waiting to start the movement. So the DBS is certainly affecting the firing of this neuron, but the neuron still pauses, has a large pause in firing right around the time that it originally paused. So this is task-related information. This is information that this neuron is encoding about the task, and the neuron's affected by DBS, but still able to conduct this information and presumably transmit it to downstream structures. Okay, let me give you another example just to drive home the point. So this is one of those neurons. Not all neurons fired like this, but this is a neuron that is driven at very high fidelity, short latency by subthalamic nucleus stimulation. Very short latency, very, very strong driving. And so if we look at the peristimulus histogram, during DBS, virtually all of the spikes happen right at this time. There are not spikes happening at any other time. So it looks like all of the spikes are being generated and sort of induced by the stimulation. And that effect of DBS is strong and present throughout the whole experiment. So this is 800 seconds of recording, and we turned on DBS for long periods of time, turned it off, back on, off, on, and you can see this elevated firing each time we turn DBS on. During this experiment, the monkey's performing the task. So when DBS is off, again, this is another typical GPI neuron, nice big pause. This time it starts before movement onset. So what happens when we turn on this very strong driving of this GPI neuron? Well, it elevates the firing dramatically, but we still see this a dip in firing. You can't really see it very well in the raster, but there is a slight shadow here. There's a reduced probability that the cell will fire right around the time that it normally pauses. In fact, the onset latency for this response is identical under the two conditions, the DBS condition and the off DBS condition. And this is an unusual cell. This is one of those cells that's driven very, very, and trained very strongly by DBS. Not all cells were like that. And so across this large population of cells, we found that a large majority of the cells had movement-related changes in activity under both DBS and no DBS conditions. And in fact, it was interesting to see that a small fraction of neurons had movement-related activity only during DBS. I'm not sure what that means, but it's suggesting that DBS is not blocking the transmission of information. Okay, and then this is reflecting how similar are the responses under the two conditions. Do they have the same timing and shape? And we used a correlation analysis to measure that. And 2/3 of the neurons that responded, the response was very similar under the two conditions. And when they were not similar, this is compared the similarity of the response, just the correlation coefficient, similarity of response versus how strongly entraining was the effect of stimulation. And so the more strongly entraining the stimulation, the less similar the responses, right? And so all of these green dots are for highly similar responses. And they happen when the entrainment is not very strong. And so the information lesion hypothesis holds for conditions when the stimulation is very strongly entraining, but most of the effects of stimulation that we see are not this strongly entraining. They're more like all of the examples that we see down here. Okay, so I'm going to skip over that and just skip to the end. So I've told you about that we think that DBS works by blocking the transmission of pathologic activity. And I think that there's very strong evidence for that. more speculatively, Robert Rosenbaum's modeling results suggest that DBS is that blocking effect is by inducing forms of short-term depression, both in axonal conduction and synaptic failure. And we tested the idea that this blocking effect is going to actually block the encoding of task information, and we don't see evidence for that in most of the neurons. Okay, and so back to this hammer idea. Okay, so if you only have a hammer, well, maybe all this pathologic activity that we've seen evidence of in Parkinson's, there's similar forms of pathologic activity in other disorders. We know that's true for dystonia. There's evidence now of OCD and Tourette's. There's also, interestingly, there's evidence for pathologic activity and cognitive dysfunction associated with Alzheimer's. And it may be that DBS will be an effective therapy for these other disorders because DBS is very good at reducing pathologic activity. So if you've got a hammer, maybe use it. So I'd like to thank the people that did all the work. Andrew Zimnick was a wonderful student in my lab, and he's now in graduate school at Columbia. Vanessa did a lot of work also, and she's now in emergency medicine at UCLA. These are my current fellows, my collaborators, and all of this was made possible by NINDS. So thank you very much for your attention.
Refresh my memory about what happens in the operating room when the electrodes are turned on and the patient is now conscious. My understanding is that something like essential tremor, you see it immediately stop. Is this also true for Parkinson's, that you see the immediate loss of tremor, I guess, that's associated with it?
No. Well, it depends on how it's measured, right? So so there are reports that well, Parkinsonian tremor is reduced dramatically within seconds after the DBS turns on.
The reason for asking this is the timing of the data that you were speaking about is very fast.
Yes.
So
yes.
I was trying to wrap my head around the idea that these data were something that were very fast, but they were talking about changing the symptoms for something that took more time.
Yes, I agree. Right. So there in the literature you can see reports that the reduction in bradykinesia takes some time, and I question that actually. I've seen very rapid effects. Rapid improvements in akinesia and bradykinesia within seconds after the DBS is turned on. And that's both in monkeys and in humans. I did a series of functional imaging studies with DBS in human patients, and we saw very dramatic short latency responses. And that's not my area of expertise, and I know that other people could educate me on that. But this is what I've seen. Very clearly, rigidity. We were able to measure rigidity in real time, and we see a reduction in rigidity within a couple seconds.
Let me just say this is one of the clearest presentations of research that I've seen. I haven't followed this for the last 5 or 6, actually more years than that. But it was really nice and clean, with the way you presented it.
Well, I appreciate that.
And I enjoyed that.
Thank you. Other questions?
Yes, I have a comment.
Comments? Yes.
I work on epilepsy mostly, and in brain slices, hippocampus mostly, and we have observed something very similar to what you're talking about. That is axonal dysfunctional, reversible method to control axonal excitability. And this has been done in vitro. And what we find that the best frequency for this is exactly 130 hertz.
Is that
So
right?
as you stimulate at that frequency, axons nearby the electrodes actually are not able to function properly, which is very similar to what you're talking about. So I don't know if there's a similarity between hippocampal axons and these, but there might be some.
Yeah, I'd love to hear how you're measuring that, because it's
We've done a lot of experiments looking particularly at axons, and compound action potential is what we measure.
I see, right, so it's field potential.
It's pure axons, yeah.
Yeah. All right.
We've even done a disconnection of the axonal pathway from the hippocampus. So looking at the alveolus, which is the pathway along this, we can see axonal dysfunction
I see.
next to the electrode.
And so one of the interesting things about this field that I think is worth thinking about is that some neurons, some neuronal elements are better made for speed and rapid firing than others. And so when I record from a cortical neuron, say a pyramidal neuron in cortex, and I stimulate in a deep structure like the subthalamic nucleus, there's a tremendous amount of failure in the antidromic activation of that neuron. It just cannot follow I know that it's being antidromically activated because I've done the collision test, but it's not able to fire at 100 hertz. We get a tremendous amount of failure along that antidromic activation. Enough that you could say, well, wait a minute, collision's not supposed to fail. Well, in this case, 100 hertz, it fails. But in these examples that I showed you, this neuron that I showed you an example of, it's firing at almost 150 hertz. Well, it's above 100 hertz. It's firing very, very rapidly. And these neurons are just built for speed. They're very good at following. And so that may be why high frequencies are required. And I don't know, it may be that some pathways, if we want to modulate the transmission along some pathways that are built for slower speeds, like the pyramidal tract, slower frequencies would be effective.
Rob, that was really nice.
Thank you.
I'm going way back to the beginning, and you showed some power spectra of these neurons firing, and I'm just wondering if you can parse more mechanisms out of some of those characteristics, like different neurons had different peak firing rates. This one in particular, there's clearly harmonics there.
Yes.
So is that related to the shape of the action potential? Does it tell you something about the circuit? So sort of the mean frequency and these harmonics kind of struck me as
so the harmonics is because of a failure. So it's skipping a phase.
Oh, okay.
Right.
That's
so if
interesting.
you don't get a spike every cycle, then you'll have twice the cycle. Right.
Or half. I mean, actually, it tells you most of them are failing.
Oh no, right.
Yeah, most of them are failing.
Yeah.
What about the frequency peaks there, though? I mean, does that have something to do with, I don't know how specific this is to body part or anything like that. Does it have anything to do with the sort of the mechanics of the motor control?
That's, it's a good question. And I don't know. So let's see. This is tremor frequency, and it's collected from a human patient who had tremor. And so, in fact, I think that Phil Starr collected accelerometer data, and so it's available for, we could do that analysis in this data. My monkeys don't get tremor. The macaque monkeys get MPTP, and they become very Parkinsonian, except for the tremor. And so we're not in a good position to study the tremor. Tremor in some ways is an anomaly. It's a kind of outlier as far as the cluster of symptoms in Parkinson's. And so in some ways an advantage that our Parkinsonian monkeys don't have tremor, because we can study the other symptoms without a pollution of this rhythmic movement of their limbs.
I'm reminded of the work that our colleague Professor Craigo has been doing recently in his retirement period on the interaction, and he's been looking primarily in the spinal cord and the motor system, which is a much simpler system, but it's a system that has anadromic and orthodromic pulses involved in here. And I'm just curious, as Professor Craigo is working on his fingernails, if he has any idea of this. Do you see anything like this similar to your work?
I took lots of notes. I took lots of notes.
Oh, I think here he is.
Okay, yeah. Well, I hope we can talk. I'd like to hear your thoughts.
So in some of the cartoons you showed of the stimulation pulse, it looked like a biphasic pulse that was symmetric.
Yes.
Is that accurate? Is that the kind of stimulation pulse you're using? Because the DBS stimulation pulse looks a little different than that.
Yes, yes. Very good. Yes, you're right. And so our stimulation was not a perfect replica of what's used clinically. You're right. So we use a balanced biphasic pulse, and yeah, and the other way in which our work differs from what's done clinically is we don't use a scaled down version of the clinical electrode, so that the shape of the contacts is different than what's used clinically. So the volume of tissue activated and the sort of work that Cameron does, clearly it doesn't. It differs somewhat from what's done clinically and in animals using those scaled-down electrodes. I can say that our results are quite similar in, say, the population activities and the types of responses that we see and those that are seen in animals that have received the more stereotyped clinical stimulation.
That's it.
All right, thank you.
Thank you very much. We do have a